Hi, my name is Nimrod Rappoport. I'm a PhD student at Tel Aviv University, advised by Ron Shamir and Amos Tanai. I'm going to present a work I did together with Roy Safra and Ron Shamir. We know that cancer is a heterogeneous disease, and it is heterogeneous even within a tissue. Traditionally, our understanding of cancer biology and therapeutic decisions were based mostly on the presence of a single biomarker, such as the expression of a single protein, as shown here. But in the last couple of decades, we used omic data to better understand cancer biology and specifically to define cancer subtypes. So given omic measurement for multiple tumors, we could apply clustering to this data and this way define cancer subtypes. In the last few years, thanks to large consortia such as TCGA, we now have not only single omic for tumors, but multi-omic data. And the question is, can we use this data to better define cancer subtypes? There were a few algorithms that were developed for this task, for example, SNF and NEMO, which we developed previously. And these methods, as well as others, work under the assumption that there is some common structure between the omics. But this assumption does not always hold, and I will show you what I mean here using an example. Here we see breast cancer data from TCGA, and we see three different omics: gene expression, DNA methylation, and microRNA expression, and we see t-SNP plots of each omic separately. Every point is a tumor, and they have the same coloring across the three different panels. So here, for example, you can see the yellow cluster, which is made of tumors that are similar in terms of their microRNA expression profile, but not very similar in terms of their gene expression profile, and not similar at all in terms of their DNA methylation profile. So we see that samples that are similar in one omic are not necessarily similar in another. We developed MONET, which is an algorithm that uses this observation, and I will show you what MONET's outputs look like. So instead of finding cancer subtypes, MONET outputs what we call patient modules. And here you see an example for a patient module, module alpha. It has three different patients, these black dots, and this module is covered, this is the terminology that we use, it is covered only by a subset of the omics. And so here it is covered by the red omic and the green omic. And what this means is that these three samples are only similar to one another in the red and in the green omic. Here we see another example of a module, module beta. It also has three different patients, and they are only similar to one another in terms of the blue omic. A patient sample can only appear at one module, at most at one module, but an omic can cover any number of modules. So this module beta could have also been covered by the red omic, for example. I will now explain what the algorithm looks like. So first we build a graph for every omic separately, in which the nodes are samples. Then we create some initial solution, and the rest of the algorithm is iterative. In every iteration, we try many different actions to improve our current solution. We try different actions, and we choose the one that best improves the solution greedily, and we stop when we can no longer improve our solution. So let's dive into what we do in every iteration. Let's say that at the beginning of the iteration, the situation looks like this, like the one I just showed you. We have these two modules, alpha and beta. So one set of actions that we try is moving samples between omics. So for example, we can take this sample and add it to module alpha, or we can take this sample and remove it from module alpha, or we can take this sample and move it from module alpha to module beta. So these actions involve moving samples between modules, but another set of actions involve changing the omics that cover the different modules. So for example, here we now added the red omic to module beta, and here we remove the green omic from module alpha. And like I said, we repeat this process in every iteration until we can no longer improve our solution. So now for the results, we applied MONET first to 10 different cancer types in TCGA, and we used three different omics: gene expression, DNA methylation, and microRNA expression. And across all the different cancer types, we look at the omics that were covering the different modules, and we see all possible subsets of the omics covering some module, which highlights the disagreement between omics, which I mentioned previously. We next compared MONET to different algorithms, and we used two main assessment criteria. One of them is the number of cancer types, in which we found a significant differences in survival in the case of MONET between the patient modules, and in the case of the other algorithms between the cancer subtypes. And we see MONET's performance here, and it is tied together with NEMO, which is the another algorithm that we developed previously. The second assessment criterion that we used is the number of cancer type, in which we found a significant enrichment of some known clinical labels. And again, you see that MONET has the best performance together with NEMO. But importantly, NEMO and MONET performed well in other cancer types, so these two methods can be used complementarily. We next look at MONET's solution on ovarian cancer. So MONET found 4 different modules. They all cover the gene expression omic, but only module number 1 covered the microRNA expression omic. MONET found a significant difference in survival between the modules, and it was the only method that we used that found such a difference in survival. There was an enrichment of non-clinical parameters within the modules, and we also looked at upregulated genes and microRNA in the module. So, for example, if we looked at module 1, which was the only one that covered the microRNA expression omic, we see that there are some upregulated microRNAs, including microRNA 514, which was recently shown to have a role in ovarian cancer progression. Finally, we applied MONET to single-cell multi-omic data. So we used data generated using the SCNMT method, which measures gene expression, DNA methylation, and DNA accessibility for single cells. We only used gene expression and DNA methylation, and the data is for mouse embryonic development at days 4.5 until 7.5. Here you see the t-SNP plots of the gene expression data, and here for the DNA methylation data, and every point is a single cell, and they are colored by the module that they were assigned to by MONET. And here you can see for every module, what are the omics that cover this module. So for example, if you look at this module at day 5.5, the epiblast at day 5.5, you can see that it's similar in terms of the gene expression profile and DNA methylation profile. And indeed, these two omics cover this module. But if you look, for example, at the mesoderm at the day 7.5, you can see that only the gene expression module covers it, and indeed it is similar in terms of its gene expression profile, but not similar in terms of its DNA methylation profile. So here we see that MONET highlights the disagreement between the different omics at this stage of mouse embryonic development. So to summarize, we developed MONET, which is an algorithm for multi-omics clustering, and it uses the observation that different omics have different structures. The modules that MONET the output is made of modules in which patients are only similar in a subset of the omics. The modules that MONET finds has clinical relevance in cancer, and it can also be used to cluster single cells. You can use MONET, it's available on GitHub, and it was published last year in PLOS ComBio, so you can read the full paper. I would like to thank Roy Safra, Ron Shamir, and Amos Tanai, and the funding agencies, and to thank you for listening.